Welcome. In this session, I'll be continuing discussing about applications of biotechnology in medicine. In the last session, already under this heading, we have seen genetically engineered insulin, and now in this session, I'm going to discuss about the gene therapy and molecular diagnosis. So let's begin this session with the gene therapy. So, what is this gene therapy? In simple terms, it is replacement of non-functional gene with the functional gene. So replacement of non-functional gene with the functional gene. So what we mean by this, see what happens. Some people may inherit certain kinds of genes that are not at all functional. They might have mutated and they are no more functional. So as a result, whatever the product that need to be produced from that particular gene will not be produced, and in that, okay, it shows various disorders, right? So now the technology has developed so much that we were able to identify that the location of the gene, okay, and we come to know that whether that gene is functional or non-functional. And if it is non-functional, then we have the technology to remove that non-functional gene, and in that we can place a functional gene. So that's what we say, the gene therapy. And these gene therapies are especially used to replace that non-functional genes, okay, if a child or an embryo we have diagnosed this situation, okay. So these gene therapies is a corrective method. So it's a corrective method of gene defect, of gene defect if diagnosed in a child or embryo. Okay, now we will understand this gene therapy by taking an example. Okay, so there is adenosine, right? Adenosine deaminase deficiency. So it's ADA deficiency. So that stands for adenosine deaminase. So this is an enzyme. So adenosine deaminase is an enzyme, and this enzyme plays a very important role in normal functioning of T lymphocytes. In normal functioning of T lymphocytes. I think by this time you might have already understood what these T lymphocytes and what role they are going to play in the process of immunity. Okay, we know that these T lymphocytes are going to involve in cell mediated immunity. Okay, so if these T lymphocytes are not properly working, then it results in decrease in the immunity of the person, right? Now we know that these enzymes are coded by the genes, and at the same, this adenosine deaminase is also coded by a gene. Now this gene is not working, so the gene that's coding for adenosine deaminase is not working. If it is not working, then the adenosine deaminase will not be produced, and if it is the case, then there will be, okay, non-functional T lymphocytes. So because I told you that this enzyme is going to play a very important role in the normal functioning of the T lymphocytes. So if I say that uh, the gene, say it's got mutated, and this mutated gene is going to, of course, will not get expressed, so hence no adenosine deaminase enzyme. So there will be no adenosine deaminase enzyme. So if that enzyme is not present, then the T lymphocytes, okay, do not function normally. So as a result, the person with this problem will be having less immunity. Okay. Now, for the first time in 1990, okay, a child named a 4-year old girl in 1990. Okay, so this gene therapy, so that's the clinical gene therapy, clinical gene therapy was for the first time, okay, used on a 4-year old girl. So it was, okay, used on a 4-year old girl named Ashanti De Silva. So on her, they performed this gene therapy. Okay. Now here, so let's see that how they have performed this gene therapy and how they try to make it successful. I hope this is understood. So this gene, which is coding for adenosine deaminase, if it becomes mutated, then there will be no ADA, okay, enzyme, and thereby resulting in the presence of non-functional T lymphocytes. Okay, so now what they have done in this gene therapy is that they have collected some T lymphocytes from the 4-year old girl. So some T lymphocytes were collected from, okay, some T lymphocytes were collected from 4-year old girl. Okay, and of course the functional gene, or that is the gene that is coding for adenosine deaminase, we know that what they have uh, generated this gene by using the RNA. So here in the T lymphocytes, so they have come to know the gene that is coding for adenosine deaminase. Okay, say suppose this is the portion, the gene for ADA, and this one will undergo the process of okay, transcription to produce the mRNA. Okay, so by using this mRNA, I can develop DNA. That DNA that has been produced by using the mRNA is specifically called the cDNA. It's called complementary DNA molecule, and that will have the same sequences as that of this gene for the ADA. Okay, now these T lymphocytes, so they have taken these T lymphocytes, okay, from that 4-year old girl and are cultured, okay, outside our body. So see here, this is the T lymphocytes that they have or collected from, of course they collected the blood. From the blood they have isolated these T lymphocytes, or I simply say T cells, right? And now what they have done, so I told you that from these T lymphocytes, means the functional normal T lymphocytes, not from her, okay, where this gene for ADA is functional, So in in this girl that gene is not functional, so we cannot collect it from her. So we have taken, okay, from another person where in the T lymphocytes the gene for ADA is functional. Okay, on transcription we know it's going to produce mRNA, and we have collected that mRNAs, and from that we developed a DNA. So that is called complementary DNA, and that is being linked to a retroviral vector. I told you in our previous session. So for transforming the animal cells, we use these retroviral vectors. So here, so this what I say, ADA cDNA is linked to a retrovirus, is linked to retrovirus. So now that retrovirus is present here. So this is the retrovirus, and this retrovirus is the vector, okay, that has the functional gene for ADA, and these retroviruses are allowed to infect the T cells. So once these viruses enter the T cells of that girl, okay, of course this we are all performing not inside her body, outside her body. So we have collected a small, okay, quantity of blood, and from that blood we have isolated the lymphocytes, and we are growing in a culture those T lymphocytes, right? And in that culture, okay, along with the T lymphocytes we have added this retrovirus to which we have linked this functional ADA gene. Now when this retrovirus infects, okay, so it's going to enter the T cells, and then finally enters into the DNA of these T lymphocytes, and finally it gets incorporated into the T cell, okay. And then this, once it gets incorporated, so this functional ADA gene will be working normally. It will undergo transcription, it produces the normal mRNA, and that undergoes translation and produces the enzyme adenosine deaminase, with which the T lymphocytes will be working or functioning normally, right? So these T lymphocytes which were infected with this retrovirus, okay, vector, so once this is finished, these T lymphocytes are going to be introduced back into the sorry, right? So that is into her backboard, okay, back. So here these T lymphocytes, now they have functional ADA gene. Now this need to be performed many times, so because only a small number of T lymphocytes we have taken out, and moreover these T lymphocytes are not, immortal, means they cannot live for the rest of the life. So they are mortal, so they have like a lifespan, so they live for, okay, a few days to few months, then later they die. So hence we need to perform this multiple times. So take some quantity of blood from her, then isolate these lymphocytes, and then, okay, infect those lymphocytes with these retroviruses carrying the functional ADA, and then put back these T lymphocytes into her body so that she can have the normal immunity. So this is how we have performed this gene therapy. But the point is that already she has grown up. Now, but if this same procedure, if we are able to do it in the embryonic conditions, okay, then this can be a permanent cure. So no need to take cells again and again, and then use this process, okay. So such things need not be. So if we are able to deliver that non-functional replace that non-functional gene with the functional gene in the embryonic conditions, so we need to diagnose any gene defects in the embryonic stages itself, And once we come to know that if there are any gene defects, then those defect genes need to be replaced with the functional gene. So of course, most of these procedures are okay in their initial stages, and slowly they are developing in future. Okay, not that far future, so in very near future we can have different methods in medicine that the doctors can come to know at an early stage that there are certain defects in the genes in the developing embryo, and they can even replace that defective genes with the normal functional genes. So that's all about this gene therapy. And one more point, so as we are replacing the gene here, sorry, enzyme, replacing the enzyme, so hence you can also call this one especially as enzyme replacement therapy. So you can use another okay term for this enzyme replacement okay therapy for that ADA deficiency. Anyway, so next we'll see about molecular diagnosis. So molecular diagnosis. So here, what is this molecular diagnosis? See, uh, at present scenario, so a person when he starts showing the symptoms means he has some disease. Okay, so when he starts showing the symptoms, then only he goes to doctor, and doctor asks to go for some tests. Okay, so that may be a blood test, okay, urine analysis, and so on. So and then in doing so they can come to know that what disease the person is having. And but this condition is only after the person already came to a point in the disease. Now biotechnology is promising that in future they are going to develop techniques that can help the persons to detect even before they get disease. So that so in after a few days or of months, okay, I'm going to have some certain kind of disease. So point is that if we are able to detect a particular disease much earlier, then it will be very easy to cure the disease, rather than the disease has gone to certain number. I mean, what you say, certain days have already passed, okay, having that disease. And so uh, in that case, treating that disease becomes very quite difficult. So I think even in human health and diseases, you might have understood that if cancers are detected early, then it's very easy, you can completely cure, permanently cure cancer. But if it been past number of days having that problem, or that is cancer, it's very quite difficult to treat, okay, in advanced stages. So in early stages, diseases can be easily cured. So that is what we will be studying under this molecular diagnosis. So it will help us to identify diseases much earlier, so that makes us easy to treat those diseases, okay, rather than in advanced stages, right? So here we have conventional methods of diagnosis. Conventional methods of diagnosis, that's just now I have mentioned some of these conventional methods of diagnosis, like the serum analysis, and then urine analysis, so etc. All these are conventional methods of diagnosis here, right? And with these methods of diagnosis, Okay, early detection is not possible. So early detection of disease is not possible. So here the point is, you know, the point, the time at which a particular pathogen entered into your body, and the time when you start showing symptoms. So there will be a gap. So different organisms will have a different time period. So what you call it incubation periods. Okay, so like say suppose in case of malaria it may be 14 days. Okay, so approximately which I'm telling. Okay, and there are different species will have different incubation periods. Different pathogens will have different incubation periods. So even you know what is the incubation period in case of COVID, right? So it means that when a person starts showing symptoms, so already that pathogen number has increased in his or her body. So that is the reason why the person shows. So when it enters, there will be no symptoms shown. So once that pathogen, after entering into the body, it starts multiplying, becoming more in number, and when your immunity system fails to stop its multiplication, then you start showing symptoms. By this time already, the pathogen's number is very high, right? So uh, in that case, that is the advanced, almost to the advanced stage they have come up, and treating at that stages is quite difficult. So what scientists say is that if a particular disease is diagnosed at its early stage, then it will be quite easier to treat those diseases. So for that reason, so biotechnology is promising to find these diseases at an early, okay, stages. Now, the methods like rDNA, so recombinant, okay, DNA technology. So rDNA technology, then PCR, and one more called ELISA, that stands for enzyme linked immunosorbent assay. So it's called enzyme linked immunosorbent assay. So these are three methods that can be used to detect disease at early, okay, stages. So are used to detect diseases at early stages. So detect diseases at early stages. So rDNA technologies, they can be used, and PCR, so that is nothing but the polymerase chain reaction. So that can also be, okay, used, so to find out the disease at the early stages. So now let's look at these techniques, how they can be used to find out the diseases at early stages, right? So let's see these techniques, right? So here, amplification of bacterial or viral. So amplification of this bacterial or viral DNAs by using PCR. So when these viruses or bacteria, when they entered into our body, so in the beginning their concentration will be very less. So their number will be very less. Okay, and gradually as the time passes, so I told you within that onset of the symptoms. Okay, so when we show the onset of symptoms, while by the time the number or the concentration of that particular pathogen already increased. Okay, that time the person will be showing the symptoms. Now by using this PCR, we can detect, so we can detect the smallest quantity of that bacteria or viruses, right? So that's amplification of this bacterial or viral DNAs or viral genomes. So we can consider any of right. Okay, right. So the genomes, I mean either DNAs or RNAs also. So I can say nucleic acids. So amplification of bacterial or viral nucleic acids by using PCR. So not just I'll mention DNA. Okay, nucleic acids by using PCR. By using PCR. Okay, right, we know already. So the PCR will help to amplify to billions of copies. So here if you take the sample from the person and amplify by using PCR, so we can come to know that with what the person is suffering from. So this PCR is extensively used in, okay, used in detecting HIV, is detecting HIV. Okay, so here in the initial days of HIV, so the person will be having a very very low concentration of this virus. So by amplifying those virus, nucleic acids by using PCR, we can come to know that whether the person is having HIV or not in the early stages. So that is what the PCR is extensively used in detecting these HIV like diseases, right? And we can also detect you know the mutations in case of cancer patients, right? So can also be used to detect mutations in cancer patients. Okay, so how they are going to detect mutations in the okay, in the genes of the cancer patients, right? So what they are going to do in this case, they will be taking a radioactive labeled, okay, single stranded DNA or RNA. So that is single stranded DNA or RNA they are going to take. So these single stranded DNAs or RNAs are radioactive. So they are radioactive, and these radioactive labeled, okay, single stranded DNAs or RNAs are called probes. So they are called probes. Now, in case of cancer patients, we know that certain genes that used to control, okay, cell division. So the genes, there will be genes that will be controlling the cell division. Okay, say suppose here is a gene on the DNA, like right. So this is a gene that's controlling the cell division, and now what happened, a mutation has taken place in this gene. As a result, the cell division became uncontrollable. Okay, that's what happens in case of cancer. Now what I'm going to do is we'll take a DNA, single stranded DNA molecule. So this is DNA, and this is say suppose a gene, so that is mutated. Okay, say here I'm going to take a T C G G A T G C, and say here this has mutated. So in that place, what we have is another base. So in this case, say suppose it has again C. So this is a point, okay, the mutation has occurred. Now this is the DNA probe that is radioactively labeled. So that is radioactively labeled. Now when I allow this DNA probe that is radioactively labeled and it has correct sequences, it is not mutated here. Okay, so when it is allowed to form a complementary with this gene, it cannot form. Understood? So what happens? Okay, so that gene will not be shown in the photographic film. So here that's what once this is finished, we are going to do autoradiography or photographic film. So once we produce that photographic film, on the photographic film we will not see this gene, because as it is mutated, this DNA probe was not able to bind because it is not having that complementary, and that is not seen in case of that photographic film. So if it is the case, then we will identify that uh, gene is mutated. Okay, so once we perform that photographic, of course the same process of separating by using electrophores and all, so the band patterns with that, so there is a lot process, even though I have told it okay here, so that has a lot a bigger process, right? So say this is the photographic film, on that a particular segment will not be seen, so that is the gene which is mutated. Okay, so it will not form the complementary with the mutated gene. So this is how even we can detect the mutations in the cancer patients. Right, with this I'll end today's session. So we'll see some more points on this molecular diagnosis in the next session, and I'll also discuss about ELISA, how they are going to perform. We'll see a small example in that case. Of course there are varieties of ELISAs, techniques. We'll see one small example in that, how they are going to detect these antigens, antibodies, or proteins or glycoproteins by using this ELISA. Okay, uh, we'll see that in the next session.